question answered, you have to work a bit for it. So I can hear and see you actually want the question answered.
So it's really exciting to be able to host you, Cindy, and hand over to you.
Thank you, Ben, for the introduction and inviting me. I've never been to Bristol before, so I'm very happy to be here. I'm very happy to see everybody online and everybody in the room. So, is it working? No. Yeah. Yes. So what is this talk about? So because I'm very happy to be here and I'm presenting about my two favorite topics, which is of course genetics and genomics and dinosaurs. So yeah, it's also about osteoarthritis, of course, right? Osteoarthritis is one of the most common diseases worldwide, as we know. It's a complex degenerative disease, and it's a disease of the whole joint. So it affects multiple tissues within the joint. And coming back to my first slide, it's actually also one of the oldest known diseases in the world, because we have fossilized evidence of osteoarthritis, which makes it even more strange if I tell you that we have currently no curative treatment for osteoarthritis. The only treatments we have are symptom management. So this is why I do osteoarthritis research. And I already told you that osteoarthritis is a complex disease. Then you change something and I can't correct. Sorry. So osteoarthritis is a complex disease, and for those who might not know what I mean by that, not only it might be complex to study, but what I mean is that in the scientific sense of the term is that osteoarthritis risk is determined by both environmental and genetic factors. So how we know, such as age, obesity, but also the genetics. And I am very interested in the genetics, like I said in the beginning. And for osteoarthritis, this genetic part is actually quite large. It's between 39 and 60% of the risk is determined by the genetics. And it depends a bit on which joint. So this is what I want to study. In your genetics, we look at the genetic variations. So how do we do this? Well, we do this through a genome-wide association study. So very simply put, I take a lot of people and I group them into two groups, one with osteoarthritis and one without. And then I have the computer tell me which genetic variations occur more or only in people with osteoarthritis. So, and the idea of why you should do this is that we can do this to identify genes that are involved in the pathology of osteoarthritis. And not only just involved in the pathology, they might actually show us the causal pathology, because you're born with your genetics. They underlie your risk of the disease. And of course, if we know the causal, then we could also think about maybe potential treatment for osteoarthritis, some new treatment targets. So this is the idea behind doing a genome-wide association study. And as Ben said in the introduction, I've been part of this, very fortunate to be part of the Genetics of Osteoarthritis consortium. This is a summary of our first paper, which is very big. As you can see, 800,000 people where we studied the genetics of osteoarthritis. And recently we have followed this up because what you do is you make the data bigger. So you study more people. So we have about 2 million people that we studied. Where we have, so these are also the phenotypes that we studied. So these are 11 different phenotypes that we all checked the genetics of in these 2 million people in total that we had. This is how the results look like. So this is called a Manhattan plot. This is almost a GWAS plot. So what you see here on the x-axis are the chromosomes, and on the y-axis is the p-value, so the association, and every peak. So the higher the peak, the more likely your variant is associated, because every dot is a variant. And you can see this red dotted line. That one says this is the significance threshold. So everything above it is significant. So that's a lot. So if I plot this in a different way, I have a little person here. I have all the variants that are associated to every phenotype here. Maybe a bit more easier to see is if I put it in numbers. But we have 962 variants identified associated with osteoarthritis. If you look across the phenotypes, there's some overlap, so you have 339 unique ones. So that's cool, right? For the ones of you that might have paid attention or I have now completely lost, I'm not talking about genes here, right? I don't talk about genes, I'm not talking about treatment. I showed you these variants and numbers, big numbers, but I'm not showing you genes. So, and this is actually for genetics, if we look closer, so this is a zoom in of one of those Manhattan plots. And I circled this little dot here. This dot is what you would see the peak of those big lines in the Manhattan plot. We call this the lead variant. So this is one GWAS variant. But what lots of people might not realize is that this is a signal, which means it's not that one variant, but it's all these variants, those that are associated with osteoarthritis. And as you can see in the plot, if I go down on this, that's a whole bunch of genes are underlying that. So any one of those genes could actually be my gene that is influencing my phenotype. And it's not even only those genes, it can be genes even further away, because we know gene regulatory elements can regulate genes quite far away. And to even make it more complex, I can even have two GWAS signals very close to each other. So do they map to the same gene or to different genes? So basically to say, GWASs are actually really bad at identifying genes. Even though all of us geneticists and GWAS researchers say, oh, we're doing GWAS to identify new genes involved in the disease. No, they're actually not very good at it. You need to think about how to do this. How you have to keep in mind these variants, these signals, there are multiple variants there. And you have to think, okay, what gene could be actually there? Fortunately, we do have methods for that. So we have quite some integrative translational genomics or functional genomics to do this. So, and don't worry, I'm going to walk you through this. So if I go back to this super complex figure with all these names and numbers in it, so these are the variants, I'm going to zoom in on one, that one to be precise. And that one is actually an old one, because that one has been found before with hand osteoarthritis. So this is an old GWAS I once did in 2016, almost 10 years ago, where I looked at the severity of hand osteoarthritis and I studied it. And then you get a Manhattan plot like this. This is much easier to look at, just one peak, sometimes called the Dubai plot, just one big tower. You can see there on the top, there's this top variant. You can see the numbers below. You see the significance. And of course, in a GWAS, you replicate. So you see it's replicated. There's this one variant. And if I look at this one variant more closely, so this is a schematic representation of a zoom in. So what you see, you have the genes on top. I colored it in. And at the bottom, you have all these lines. Those are the variants. Those are all the variants in my GWAS signal that are linked to hand osteoarthritis. And this is called the LD block. So this whole block, any of those genes could be, and also outside of it, any of those genes could be my causal genes. So the first thing you can do is you can look, okay, what do these variants do? Are there perhaps variants in there that might affect the protein structure of my gene? Fortunately, in my case, the answer was yes. So I have one that does that. And I also have some that are in the untranslated regions of the gene. So they are in the 5' 3' UTR that might have something to do with gene regulation. So so this already goes, oh, well, maybe, you know, this MGP gene is interesting. So just for the sake of this presentation, I've not shown you all the results, but actually in some of these other genes nearby, we also have variants that are in the coding sequence of other genes. So it's not always that easy. So, but what we can do and what we did is, because if you have a coding variant, think about, in genetics, you have two alleles. You get one set of genes from your dad, one set of genes from your mom. So you have two alleles. It means you have two copies of a gene. And if you usually, for most of the genes you have, you'll see that the gene expression level from each gene is has a ratio of 50-50. So each allele will express the gene the same amount. But you can imagine maybe in one of those alleles, I have a certain variant. And what can happen is that variant might affect the amount of gene expression you have. This is called allelic imbalance. So maybe because you have the variant, you have either less or more expression of that gene, which indicates that that variant might actually be related to that gene and that gene related to your phenotype. So we did this for matrix GLA protein, or this MGP gene. And what we find is this, in this little table, you can see the line indicates the 50-50 ratio is what you would expect. And you see if you have the variant, everybody's below it. And this is really nice. So you see, if you have this variant, which increases your risk for osteoarthritis, you have less MGP expression. That's nice. That's interesting. So what does it do then? Is MGP a gene that's interesting to look at? Well, actually, yes, it is. MGP is essential for the inhibition of calcification. So in your joints, your cartilage can be calcified and it can then break down, which might then lead to osteoarthritis. So that's really nice. That's a nice loop from the GWAS. We do get a gene. But if we go a bit further, we can see that MGP can only inhibit cartilage calcification if it's been gamma carboxylized, you can forget the hard word, by vitamin K, which basically means it needs to be activated by vitamin K. That's interesting because vitamin K has lots of interesting functions. The main one of which we know that vitamin K is involved in anticoagulation, in blood coagulation. The same proteins that involved for the clotting of your blood need the same activation as MGP does. So interestingly, lots of people take anticoagulant medication that actually inhibits this vitamin K pathway. And if my genetics identifies underlying causal pathways for osteoarthritis, and this gene is really important, then inhibiting this vitamin K pathway inhibits that gene, so you have less of that gene, less active of that gene, you would have a higher risk of osteoarthritis, right? So the cool thing is I don't only do genetics, I also do epidemiological studies. And I have a really nice data set where I can look at that. It's called the Rotterdam study. I have quite some data and I have follow-ups. So I have 5 and 10-year follow-ups of these people. I have X-rays and all sorts of other data, including pharmacogenetics, genomics data. So I know what kind of medication they're taking and I know how much they're taking. So let's see what happens. Well, if we look at the people who take these vitamin K inhibitors versus people who don't take them, you can see you have a significantly increased risk of getting osteoarthritis. So I made a table and the figures are the same. So in the colored bar, those are the people who take the vitamin K inhibiting medication. In the non-colored bar, those are the reference group. So you can see there's a significantly increased risk for both hip and knee osteoarthritis. So that's pretty cool. So maybe the clinicians under you will immediately think, confounding by indication. So confounding by indication means that I compare people who take the medication versus people who don't take the medication. But the people who take the medication, they take it for a reason. And actually, most people who take these anticoagulant drugs, actually, they have atrial fibrillation, so heart problems. So what could also mean is that maybe it's not the inhibiting of vitamin K that leads to this result that I see, but it's actually because the underlying cause. These people have a certain disease, and this disease leads them to having a higher risk of osteoarthritis. So that's a bit of a problem. But the nice thing is you also have another set of medications that are also anticoagulants, but don't inhibit vitamin K. So if we think it is actually the underlying disease that causes the association that I see, you would expect that both the vitamin K inhibiting anticoagulants and the non-vitamin K inhibiting anticoagulants would both result in an association. But if it really is because of the inhibiting of vitamin K, I should only see this effect in the vitamin K inhibiting medication and not in the others. So that's a way to solve it. And fortunately, I could solve it because we collaborated with people who had a really nice data set on people who take these direct oral anticoagulants, so non-vitamin K inhibitors, and they compared this group of people to people who take vitamin K inhibitors. And what you see in the vitamin K inhibiting group people, so that's warfarin, they had a significantly increased risk of getting a total joint replacement, the end stage part of osteoarthritis. So that's quite nice, right? We have now, we had a genetic hit, we have a pathway, we have a gene, and we actually show now that this pathway is important. We find a causal pathway. But I said genetic hit, right? So what about the genetics? So I haven't looked at genetics in this story. If, because you had a genetic variant that caused less MGP gene expression, what would happen if somebody who has that genetic variant, who already then has less MGP, and then gets their vitamin K inhibited? Wouldn't they maybe get an even higher risk? They get a sort of double effect. Next to that, we also know that for this particular drug, If you have a certain genetic background, you need a much higher doses. But this dosage is based on your functioning of these proteins in the liver. Maybe this is a much too high dose for other tissues. So we check that. And the nice thing is what you see. Again, the colored bars are the people who take the vitamin K inhibiting drugs. And the ones all the way at the end, the highest one, that's the group of people who are carriers for this MGP risk variant. So they have less MGP and are the people who take the highest dose of this drug. And you can see, yes, indeed, they have the highest risk for osteoarthritis. So that's really nice. That's pretty cool. So, however, I said, I can find causal pathways with this, right? And I said MGP inhibits calcification. And I just looked only at the vitamin K pathway, not at this calcification part. Because if I see very strong evidence that inhibiting vitamin K is detrimental, that means that this, the thing that MGP inhibits is also important, which is the chondrocalcinosis or the calcification. So, and we actually know, if you look at literature, that this calcification occurs a lot in osteoarthritis, but we mainly see it in end stage. It's also linked to calcium pyrophosphate depositing disease. Sorry. So maybe there's something there as well. So why am I doing all this vitamin K stuff? I can just measure chondrocalcinosis. I have X-rays from people, and I can measure in these X-rays. I can see this clouding. I highlighted it here. I highlighted it because I don't see it, so somebody else can score it. And then we can see, look, is there then also a link between this chondrocalcinosis and osteoarthritis, right? So again, we have this nice data set of the Rotterdam study. I have this follow-up data, and I have a very nice younger brother who's a radiologist and scored 30,000 X-rays for me. So then I have this data, so then I can actually study it. So, and that's what we did. So we looked to see if chondrocalcinosis, if you have it, increases your risk of getting osteoarthritis. And yes, it does. And we also replicated this. So we have two independent cohorts. One is the Rotterdam study. One is the MOST cohort in the US. And we see this in both cohorts. Even more so, if you now look below the red circled bar that I have here, we also measured the severity of the chondrocalcinosis, not just the presence, but also the severity. And indeed, you see, the more severe it is, the higher your risk of getting osteoarthritis. So this is really great, right? I now have my pathways. So I might be hearing you thinking, Cindy, if this is all causal, what about the dinosaur? Right? The dinosaur had osteoarthritis as well. Well, yes, the dinosaurs also had chondrocalcinosis. So I even have validation here. No, but in all seriousness, what does this mean? This means that definitely, in my view, this is an important pathway. We can maybe actually use this. For instance, in the Netherlands, we are talking about guideline changes for people who maybe already have osteoarthritis and they need anticoagulants to first see if they can have non-vitamin K inhibiting anticoagulants to either prevent the severity of their osteoarthritis or in other risk group to not give them that, so they have to prevent osteoarthritis. The other one is maybe we should screen people for the MGP genetics before we give them vitamin K inhibitants. That's another option. Another one is, well, if this vitamin K is that important, what about a clinical trial? Could vitamin K be used as a therapeutic target, for example? Could we give people vitamin K? Does that make sense? The same for calcification. Should we maybe do something there? If we see people with chondrocalcinosis, should we perhaps treat them or do something to prevent osteoarthritis? So for the trial part, I'm actually working on that to see if molecularly, if we give people vitamin K orally, does that actually do something with their molecular effects in the cartilage? So I take people who are on a waiting list for a total joint replacement of the knee. I give them 5 weeks of high doses of vitamin K. And then I will measure the MGP activation in the blood and in the joint, and also other omics measures to see, well, do we get the omics profile that is protective of OA rather than damaging. And what's more really interesting here is because maybe some of you would be saying, vitamins, what's that going to do? And I will tell you absolutely true, because most vitamins, they have a sort of threshold. You can, for instance, you can take a lot of vitamin C. If you have enough, you have enough. More is not going to help. The same is probably true for vitamin K. And then it's very interesting to know that 60% of the people over 60 have a subclinical deficiency of vitamin K. So a clinical deficiency would mean you have problems with your blood coagulations. But this group of people have a very low level of vitamin K, which is enough to make sure that your blood coagulation works well, but might have other effects on the other tissues and the other functions of vitamin K. So there might be a group of people that might actually, have benefit of this. And I'm hoping to know the answer to this question in 2 years' time. So we all need to have a little bit of patience. So, but I was talking about genetics, and I was talking about all these genetics that I found. And I've now just spent a lot of time about one variant. And I've shown you all these effects this one variant has caused. We found a new pathway. We found a possible clinical route we can take. But this is one variant. There's 962, right? So there's 961 others that we can look at and do something with and might actually really have an impact on osteoarthritis research and in the clinic for the patient. So fortunately, some of you might be thinking, oh, there was a lot of work you did there, Cindy. We don't know the genes yet. So fortunately, in the Genetics of Osteoarthritis consortium, we agreed with that. So we actually did a lot of this work already in that paper. So we use what we call lines of evidence. So for instance, this allele-specific expression that I showed you, but also things as EQTLs, which you look at the how a genetic variant could alter the expression of a certain gene, and many others. I listed some here, that we use them to give you an idea of what could be the actual effector gene within the GWAS locus. So that's in the paper. And we then say, look, if a gene has 3 or more of those lines of evidence, we call it, this is a possible good candidate. And we have 700 of those. It's still a lot, but we can start looking, okay, we already have a list to start from and to work from. And if we look at that in detail, we can actually start to see some pathways occurring. So these are broad pathways. They might not be as super defined as the MGP and vitamin K, but these are really good pathways to start with, to go look into more detail. So as you can see, for some of these pathways, we have a lot of genes in there. And novel genes means the additional new genes in that pathway we found versus our first study and now the new study. But you can see there's quite some there that we could use. So this is a question I get a lot. So I already know my gene. What does the genetics do for me? What I mean by that is there's lots and lots of researchers that do actual wet lab research, and they usually have genes that they're interested and already working with. And the work that they do is really important because all the things I just showed you have not been validated in the wet lab. As long as they're not validated, they're not really the true effector gene. So we need to validate these. So this is the first thing, right? So we need the validation. So all these people already working on genes, they can select new ones or check if their own gene is already in that list. But why would you do that? Well, you would do it for this reason. We know that if you design a certain treatment or a drug, and your drug or the gene it's based on has a genetic association, genetic line of evidence with the disease, your drug is almost 2 to 4-fold more likely to actually successfully pass clinical trials. So these are some big studies from the FDA. So again, this is this causality that I talked about in the beginning. Genetics can identify underlying causality. So even if you know your gene, this is something you can do. Then the other question, so we've done this also parts of it. So parts of this, from all the genes we identified, we looked, well, are there already existing drugs that target those genes? And it's a really big list, as you can see, that's quite a large number. The only problem here is, I will be honest about this, is that for only 2% of all these genes and variants that we find, we know the link between the gene variant, the genetic variant and the gene. And then very importantly, we know if the genetic variant increases or decreases the gene in expression. So that's very important to know, right? You can maybe say, oh, this drug is useful for drug repurposing, but if it has the opposite effect, it would actually increase the risk of OA instead of decreasing it. So this is a gap in knowledge that I and myself and others are currently working on to see how to address this, but unfortunately it's not too easy. Fortunately, I then have a job to do. So the other thing is, so what does genetics do for the clinic? Well, I gave you an example with the MGP, so it might actually have real clinical implications there. Another one, which lots of people mention, is polygenetic risk scores, right? So you can use all these genetic variants to predict somebody's disease risk, at least the genetic part of the disease risk. So we've also done studies on this, and most people will know it works. You see an association, but is this better than just doing age, sex, and BMI? No, it's not, right? So how does it really have a purpose as a prediction? Maybe not, but you can also use it in a different way. You can also look at these people which are in this high risk group, for example, or better yet, you can actually look in people who already have osteoarthritis. If you look in people who already have osteoarthritis and see what their genetic risk factors are, so what are their underlying causal risk factors, you could see maybe genetically which pathway is the one that underlies their osteoarthritis. Osteoarthritis is a multifactorial disease, many different pathways are involved. So for example, maybe I have somebody with osteoarthritis and I see, this person has this MGP variant, and I would say many others as well that are in the same pathway. I can say, your OA, at least from the genetic point of view, is in part caused by issues in this pathway. Maybe I should give you vitamin K or another drug. So you can go into personalized medicines with these PRSs without actually using them for prediction. The only downside here is we have to understand the pathways. Like, what do these genetic variants do? So which pathways are they linked? How do they work? So this is why it's also really important to validate those genes and really find what's going on. So this was my talk. So in summary, I hope you all agree that population genomics is cool. Just as osteoarthritis, very interesting. But my key message I wanted to make is that you can really use this genetics to identify drug targets and causal mechanisms. But it's a lot of hard work, so you need lots of collaboration, people with different skill sets come together to make this happen. But I think this is a very important step, and we should not only do it for osteoarthritis, that's why it's called an osteoarthritis story, because I work in osteoarthritis, but you can do this for any disease. So at least any complex disease with a genetic background. So with that, I'd like to thank all my collaborators, including my brother. And I would like to end this meeting with a picture, a bad picture of myself with a dinosaur that has osteoarthritis.
Thank you, Cindy. That was excellent. A really, really brilliant overview, and lots of emoji claps on the screen. Thank you. Just going to ask if anyone in the room has any questions, and if anybody online wants to put any questions in the chat or put their hand up, or we can allow you to unmute yourself and ask them. We'll be able to hear you in the room. Any questions? George.
So I guess you obviously can't genotype dinosaurs, but I wonder, are other sort of animal models, are there good other animal models for osteoarthritis? For example, do mice work as a model for osteoarthritis?
So I'm not an animal model person. I know lots of people that work on animal models of osteoarthritis. So I know mice are used a lot, but also larger animals to really look at the joint loading, such as goats, for instance. I know people who really work in those models to really specifically target tissue regeneration, for example, which could be a very good way to maybe treat osteoarthritis. But no, I wouldn't know. So to be honest, I don't work with animal models. I don't want to alienate everybody, but since I'm interested in helping humans, I'd like to see if I can focus all my research in humans. And I think nowadays we can do this really well. For instance, one of the projects I am involved in, it's called iCell. It's led by my PI. It's called Professor Joyce van Merks. And what we do there is we use iPSC cells. iPSC cells, you can so induce pluripotent stem cells. You can culture them and differentiate them into other cell types. For instance, cartilage or bone, whatever. The nice thing about this model is it's a very genetic model, in the sense that if you have different iPSC cell lines, they might show different rates of differentiation or different gene expression, for example. It's mostly based in their genetics. And what we can do now is we can actually, in a single plate, not culture one cell line, but 80. So 80 individual iPSC cell line representing a population in one experiment, and I can differentiate them, for instance, do cartilage, give them a certain treatment, or a certain loading, for example, to see what's going on with the gene expression in the cells, because I can do single cell readout to see what's happening in every cell. So I would even say we should move to models like that over some animal models. But again, I'm not an expert, so maybe I see some glaring gaps that I've not addressed.
So with the what cell type do you differentiate them to?
Yeah, that's a good question, right? So the question is, what cell types would you differentiate them to? So the easy answer would be cartilage, right? So we know cartilage is an important tissue, but osteoarthritis is a disease of the whole joint. So now more people, for instance, Ben as well, is also very interested in bone. So it's in osteophyte formation. This is not the cartilage phenotype, but this is a bone phenotype. I've also seen studies where people look at tendons. So actually in our own GWAS paper, we also see some of these variants. They looks like they show effect in tendon tissue and not in the other tissues. So I would say as many tissues as possible that relate to the joint. And even that might still be limited, because maybe there's also other more systemic factors at play.
And do you select by the genotype?
Yeah.
What's your sort of source?
Oh, the source. So this is really nice, because iPSCs are really important for lots of research. So I think that they're actually UK based as well. You have the HIPSCI, which is a universal iPSC data bank, basically. So they have healthy population-based donors that they use to create these, and so all these cell lines are standardized, created in the same way, and also molecularly characterized. So already, if you buy a cell line there, you already know the genotype, you know some basic gene expressions, you have some basic informations of the person who donated the cells. So it's a very nice standardized database. So we use that. It's of course not ideal yet, so it's mainly white ancestry UK citizens. Ideally, if you want to really talk about the population, you want people from all genetic ancestries. But that's a really good start. So this is what we're using at the moment.
How big is it? How many can you sample from?
I'm sorry.
I mean, so to choose by genotype.
No. We chose by so the origin of the iPSC cell. So for instance, you can take them from skin or from blood. So we all wanted the same origin and the same age range and an equal distribution between male and female cells. That's it. So similarly to population, no pre-selection on genetics.
John, do you have a question?
When you're the concept of trying to use genetics to come up with targets for treatment presupposes that you're trying to look at a sort of identify susceptibility and causal pathways that are common across joints.
Yes.
To what extent are these GWAS signals joint specific, or are they
Thank you for asking. So John here asked about so are these GWAS variant joint specific? And I'm very happy you asked, because I prepared a slide. This slide. So this is new data that we're doing. So we're doing a new type of GWAS that can actually show us more insight into which genetic variants overlap between different joint sites. So multi-traits. And the nice thing about this one, we can also identify variants that have opposing effects. So what you see here in this graph are the different joints that we looked at. The numbers in every box indicates the number of variants that overlap between the two phenotypes. So I can tell you now already, we're using the Genetics of Osteoarthritis data. There's only 5 variants that associate with all of these phenotypes at the same time. So that's very, very little. most are linked to 2 or 3 traits at the same time. And interestingly here, the percentages, they give the percentage of variants that are both associated with these 2 phenotypes, but in opposing directions of effect. So they, for instance, this 15% you see between hip and knee means you have 15% variants that we know that overlap between the 2 that actually have an increased risk for knee osteoarthritis and a decreased risk for hip osteoarthritis, or vice versa, which I think is very interesting. I'm not sure how that would work, but this is something in the GO1, we identified 2 of those variants, and now we have, I think the number is 65 of them that have these opposing effects between different joint sites. I'm not sure what they do, but it might be really interesting to look, because instance, like you're saying, if we want to target, when you do drug targeting, you either want a drug that, a genetic variant that maybe does something with all joints. MGP, for instance, is one of them. However, MGP is also one that has opposing effects. So interestingly, within the GWAS, this MGP variant has a increased risk for hand osteoarthritis, but a decreased risk for knee osteoarthritis, which is very weird, because if you look at the other data that I just showed you, where I effectively shut down vitamin K pathway, I see an increased risk of knee osteoarthritis. So I'm not entirely sure what's going on there, but even though the variance here is opposing effects, this pathway is still a risk pathway in the other joints. So I'm not sure if we should look at variants that overlap with everything, or it doesn't matter too much. Yes.
Just a thought on that last point, Cindy. If you had really bad hand osteoarthritis, could it be that you then don't get coded for a diagnosis of knee osteoarthritis by your doctor because they think, oh, there's already osteoarthritis diagnosed, so we don't need to add the knee to already catch, or you've got so much pain in your hand, you don't go to your doctor and say, oh, by the way, I've got knee pain as well, because you already know you've got.
This could be true with the genetics of osteoarthritis data, because there the most of our phenotypic data is based on hospital data, but we also see this in the Rotterdam study data, and this is purely X-ray based. So this is not physician, it's just some X-ray diagnosis of osteoarthritis.
Yeah,
I like the idea, but I don't think it's true in this case.
Yeah, and then I'm going to ask you a question that pushes you slightly away from osteoarthritis, because I know it'll probably be of interest to people in the room and potentially online. We've seen with Mendelian randomization this explosion in the number of papers that have been written, and there's lots of papers of dubious quality. George is pulling his hair out because of it. He has no hair left. If you had hair left,
this is why you lost all the hair.
And I know that you're sort of very passionate about rooting out bad research within the omics field. I just wonder if you could comment on the landscape as you see it at the moment, and what we as a broader research community should be doing to improve upon it.
Yeah, I think you reach a really good point here, because you're talking about the Mendelian randomization, so I haven't talked about that here at all. I am talking about all these 26 lines of evidence that I showed you, where we had gene expression, EQTLs. We do all those analysis with existing data sets. So these are either freely available data sets or data sets that other people have and already published on, and we can reuse for genetics. This is why those databases exist. They're super reusable. But now what we see, at least what I've seen, and I've actually talked to editors about this as well, is that there's a group that actually also claims to reuse this data. So they write papers where they say, hi, we downloaded data from these databases, and we are looking at new genes for osteoarthritis. So we're looking at differential gene expression. They let machine learning loose on it. And they have the new genes for osteoarthritis. However, all these papers, they follow the same formula. And what you see, you have papers that use the same data set, different authors, same figures, different results. And most glaringly is that these results, or these analysis that they've claimed to have done, were already done by the original authors of the paper, and they do not match. So these are really, and this is just based on the results. If you read those papers, you'll see that the methods are not explained. There's glaring issues, there's gaps, there's nothing you can check. There's no tables. They do this overwhelm strategy where you have maybe 7 to 10 figures, all with panels A to Z, which is insane, and sort of try to overwhelm the reviewers, because these are all published papers. I personally have a list of, I think it's now 120 papers within the osteoarthritis field that follow this pattern. And I get them also to review, and then I take the time to explain to the editor, you should not accept this. They are still sometimes accepted. It's very unfortunate. And they're still being published today. So I think as a group, what we should do as a community is, one, know that this is going on. So congratulations, you have done the first task. And two, the most important thing is that if you get those reviews, those type of reviews, be really critical about them. Or if you see those published papers, be very critical when you read them. Don't cite them, don't use them. This is what we can do. The other bigger issue is with the editors to prevent this from even going to peer review. But this is hard because it's very specialized data. So you need people who know about this, and most of our science getting more and more specialized in all fields. So yeah, it's hard.
Yeah, and we can't all do it alone because there's only so many papers you can review.
Yeah, so this is why I said 120 for osteoarthritis, because I have the same, but then with a different phenotype, and it's exactly the same as my osteoarthritis paper, only now it's prostate cancer.
Yeah.
Yeah.
Any other thoughts or questions in the room or online? Otherwise, we'll draw it to a close there. Thank you very much. Oh, is there someone in the chat? Sorry, there is. So from Fiona Watt, another question about lines of evidence for GWAS results.
Yeah.
Are some more important than others? I.e., should we just be counting them to quantify this, or weighting them in some way, or looking for membership of different groupings you gave?
Yeah, that's a very good question. Something we discussed about a lot, and actually not did just because of time and amount of data. But yes, I agree with you. What we should do with those lines of evidence is probably weigh them. Because I think not all of them are equal. So what you can broadly classify them in two, which is lines of evidence that go from the variant to the gene. For instance, this EQTL or allele-specific expression, they show really the effect of this variant on a gene. And you also have a bunch of lines of evidence that say something about the gene itself. So is this gene expressed in cartilage, yes or no? Is this gene, does it have a already existing osteoarthritis or musculoskeletal-like function or phenotype? And you can say, if we're talking from a GWAS perspective, those evidences that are from variant or gene actually weigh much larger, because you could have a locus, a region in there with your GWAS hit in it, where you have multiple genes that could have a skeletal function, but only one of them, or maybe two, but let's assume one, only one of them could be your true effector gene. So yeah, I think we should probably weigh them. How to do that in exact numbers, I don't know. I think we should study more of our data, try some things out, I would say. So yes, I would say you should probably weigh them. They're not all equal. What was the last part?
Or looking for membership of different groupings.
That's also a good point. That might also reflect a bit back to the question George mentioned here about the iPSC, in which tissues are you looking? For instance, maybe you should look per tissue what the effect is. Maybe, so we know actually from some studies from Sarah Rice, actually we have some GWAS variants with the COL-GATL2 gene that has different effects in different tissues. So yeah, maybe we should indeed group them. Good idea.
And this was another question from Fiona. Can you tell us a bit about how you were delivering vitamin K in your proof of concept study, and mitigate any risks of clotting or other adverse events associated with giving vitamin K?
Okay, so first, there are no adverse effects with giving vitamin K. You can eat really, really a lot. The worst thing that might happen if you eat really, really a lot, and then I'm talking about grams level, might be an upset stomach. That's it. There's been quite some studies that actually give vitamin K for, interestingly, for arterial calcification, because MGP The other tissue it expressed is in the artery, and it actually does the same function as in cartilage, which is prevent prevent calcification. And actually, those studies are somewhat positive on giving vitamin K. So this is the first part. So actually, you can't really get adverse effects. Plus, the dosage that we'll use is a really safe dosage. It's actually lower than some of the already existing trials that give it for 5 years, and we want to give it for 5 weeks. So that's one. The other two is how do you give it? So how we're going to give it is orally. Why orally? Well, one, this is the cheapest method there is. And two, we have this evidence that vitamin K might actually be involved in multiple joint site osteoarthritis. I've seen evidence from hand, from hip, from knee. If I give something orally, it can be systemic. So cheap, ease of access, and potential to actually affect multiple joints in the body. So this is why I want to test orally. And cheap, I mean, I calculated it. It will be 300 euros per person per year, which is very, very cheap. If this could be used, and I think it could probably be used as a preventative method for a subset of people with a specific genetic background, I think. But that's a really doable thing. So this is why I wanted to do like this. You could also say, we'll just inject it, which will be much more difficult to actually implement. Not everybody can have access to it. Plus, this might not be feasible for the hand, because in which joint are you going to inject? In the hand, or are you going to do all hand joints, right? This is why.
You'd only need to inject a couple of joints and your patients won't come back.
Also, a very good point here that Ben makes, they might not come back for treatment.
So got another question saying, thanks, very interesting talk. Have you considered characterizing your SNPs according to the characteristic of some human subpopulations, such as sex, ethnicity, post-traumatic event of the joint or underlying pathologies? And then do you also find SNPs with contradictory effect in that case as well?
Okay, so we have done genome-wide association studies in different ethnicity groups, so different genetic ancestry groups. I don't think we from the Genetics of Osteoarthritis Consortium really identified genetic variants that are very specific related to certain genetic ancestries. We do know some genetic variants just don't occur in other groups. For instance, we have genetic variants that don't occur in people with Asian ancestry. But I don't think we found specific ones with specific effects. For sex, we have. So we did also sex-specific analysis. And there we indeed show there's, I think, now 7 genetic variants. I might be wrong about this number. But at least 7 genetic variants where we know there's a different effect between men and women. So either in men or in women, it has a higher risk than in the other sex. For the opposing, I showed you here. So yeah, we find opposing, but for between sex and these others, no, we haven't looked at that because we only have 7 variants for sex differences so far. And as far as I know, not really any for genetic ancestry, but it would be interesting to see if that could be the case. But I don't think we have the data right now to look at that, unfortunately.
And here's another question. So did the variant linked to vitamin K have a larger effect size or beta than others? If it is a core gene pathway for osteoarthritis, you'd hope it is, but we normally don't see that with GWAS. We see a marginal effect across all significant variants. Was that true in your case? And if so, why did you think that is?
Okay, so this is a very good question. And this is something I use actually this MGP as an example a lot of people who are critical on GWAS going, it's only small effect sizes. Because yes, the genetic variant for MGP, if you can click on the slide here, Ben, yeah. I think.
Do we go back?
Yeah. Go back.
You've got control.
So because I think I have the beta in, because the beta is super small. It's very small, so it's a very low effect. I think it's 1.01 or something. We're almost there. And I think yes, I put it here. So here you see the beta here. So it's so if we flip it, it's 1.25. So there's something there, but it's not. No,
that's quite a high beta.
Yeah, I don't think the beta is correct here. I probably put in the wrong number there, but the beta should be really small. But the beta is really small for MGP, but you see a really big effect. Why do I think this could still be the case. This is because this is a complex disease, right? So it's not a Mendelian disease. It's not one genetic variant that's related to causing the disease. It's hundreds to thousands of genetic variants. So you can imagine if you had this one variant which is in this pathway, very little effect. But if you have 1,000 variants in this pathway, then you have a really big effect. So the effect of the variant itself doesn't really say how important this pathway is. It just pinpoints this pathway as, hey, this could be a potential causal pathway for osteoarthritis. It doesn't necessarily yet say something about the importance of the pathway. I think we could only say something like this if we know all of the genetic variation. So also keep in mind that this 952 variants we found for GO2. If you look at the variance explained, so this is the percentage of the genetic risk that we explain by the variants we know. For hip, it's the highest. And I think from the top of my head, it's around 20 to 25%. So even for hip, where we know the most, we miss 75%. So it could be that there's hundreds of thousands of other variants in this pathway. So yeah.
So how big does our GWAS need to, how big does GO3 need to be to get 100% of genetic variants?
I have no idea. So if we go for height, for example, height is also super complex. You have the giant consortium for height. I think the latest GWAS for giant was 5 or 6 million people. There they claim, I think they have 89% of variants explained there.
And the key thing with height is it's a continuous variable, not osteoarthritis, which is binary.
Yeah, so then you probably need 5 million cases plus maybe 5 to 10 million controls.
We've got a couple of cases in the room, but I don't think that's quite enough.
Well, every bit counts.
Yeah.
Every bit counts. Oh yeah, for people on the line, if there's people who are, so we are running GO3.
Twice.
Oh.
But I'm probably in the GWAS twice in the UK biobank, and I was in 23andMe, so
yeah, we've double counted you.
Yeah, I know. I know. Yeah, this will explain everything. All the weird data we have in there. Yeah, it's clear.
Sorry, you were just saying.
Yeah, I was saying that currently the group of Ellie Zaghini is planning the third genetic meta-analysis. So if you have a nice cohort and have not heard of this, contact Ali because you would probably be very much welcome to join.
Great. Well, I think that's all the questions online. And I think that's all the questions in the room.
Yeah.
And we've done our hour. So thank you again, Cindy. And thank you to everyone who joined online.
Having me.
Emoji clouds.
Let's close the meeting. Hi, everyone.